everybody, and welcome to this episode of Infection Control Matters. It's Martin Kiernan here, and I have two guests with me today from a slightly different field, it's fair to say, leading for the talk about a paper published recently in the Journal of Hospital Infection is Dr. Sarah Fieldhouse, who is by background a forensic scientist. She's an associate professor in the Health, Education, Policing and Sciences Department at the University of Staffordshire. And as I said, she comes from a background in criminology and forensic science. And my other guest is Dr. Emmanuel Babafemi, who is a senior lecturer in biomedical sciences, also at the University of Staffordshire, but he is a biomedical scientist by background with a specialism in microbiology. And so very much grateful to the pair of you for sparing some time to have a chat to me. So thank you very much.
You're welcome.
Very welcome.
Now, the paper we're going to talk about was published in Press late 2025 and is now out in the Journal of Hospital Infection. It's an open access paper called Assessing the Effectiveness of Hospital Cleaning Using Fluorescence, a proof of concept study and comparison with ATP testing. So, Sarah, can I start off? Your background's in fingerprinting and crime scenes. How did you end up? I mean, people might argue that hospitals are a crime scene at the moment, but how did you end up getting involved in a study looking at assessing hospital cleanliness?
One of my main interests in forensics has been about making invisible substances visible, sort of very aware that, in most environments we sit in, there's lots of substances that we can't see around us. And the technologies used within crime scene examination and within sort of forensic biology. But one of the problems in forensics is differentiating between what might be evidentially useful from just routine contamination, which, as I say, causes problems for that as a method, because you wouldn't necessarily be swabbing anything that fluoresced for DNA, for example.
Right. Okay.
Now, if we think about a hospital environment where you're wanting an environment to be very clean, like the presence of a substance think you'd want to remove. Certainly if visually you could see something, you'd remove it. So awareness of having lots of invisible substances there, and knowing that things like body fluids and lots of foodstuffs will fluoresce, there's a sort of logical translation there. So my academic curiosity led me to approach several NHS hospitals and speak to facilities managers or infection prevention leads, and come on the call and say, I've got a strong background in forensic science. Would you let me into your hospital to trial this method? And to be honest, Martin, a lot of people sort of looked at me like I was absolutely crazy. And then I spoke to a colleague who's somebody that I've kept in touch with and I now work very closely with. And he said, yeah, sure, come in, let's see what you can find. And so I asked him and got introduced to his facilities team and infection prevention team, who were absolutely so open-minded and welcoming. And I said, well, what do you do at the moment to assess the cleanliness of your environments? And explained that obviously visual assessments understandably govern their sort of cleaning audits. And they were using something called ATP testing, which I'm sure your listeners are familiar with. So I said, okay, well, let's see what we find, and let's use ATP testing, your environmental monitoring method, and see what we can find. And that's where the study started. And then since then, it's sort of evolved to be working with colleagues such as Emmanuel, who is helping to work out, okay, of what we're finding, what's the sort of microbial content of it? And then it's spanning out into AI and virtual reality and all sorts of fields, which again, it's a wonderful project. And it's been fantastic really to share it within the Journal of Hospital Infection, because again, that's to come from a forensics background and to be able to really interrogate that data and discuss it with experts in that field. Sort of the review process, although it was tough, it was wonderful, and I'm just honoured to have the paper and for you to invite me to this podcast.
I mean, it's something that actually always gets a lot of interest, is how clean is clean and how do we determine what's clean? Because if you look at the NHS cleaning manual, visual inspection is the way to go. Now, okay, if you see stuff, fine, but a lot of it you just don't see, which is why many of us in the
Mm.
past have used things like ATP or have even used UV dots. Now, visual inspection, as I said, I've never been that satisfied with it because it's purely qualitative. It's not, strictly speaking, quantitative. You can't really measure it. It's either your impression, it's how you feel about something often, and what some people will accept isn't necessarily what somebody else will accept. When you start moving into the area of did a UV dot get removed, it's either gone or it's not, and ATP will give you a reading. So this looks like it might give us slightly quicker feedback. And how did the results go then? Look, comparing ATP with what you found looking at fluorescence, because I actually have borrowed a fluorescent torch once before and walked around my hospital probably 15 years ago now, and it was quite horrific, to be honest. I mean, my office was fairly horrific, but the rest of the place and the toilets were spectacularly horrific. So talk
Mm.
about a comparison with, say, the ATP findings that the hospital was using and your fluorescence.
I think it's important, just before I go into the ATP, just to mention that the lights that we were using in the project are sort of forensic grade lights, and it could be very well aligned to what you used previously.
Mm.
But say the UV lights that you'd use to fluoresce UV gel markers for cleaning, these are quite
That's
different.
different,
The
isn't it? Yeah.
intensity of the light,
Yeah.
it is. And it's also, we use UV light, but we also use blue light.
Mm-hmm.
Blue light tends to be applied in forensic science to have a broader sort of fluoresces, a broader range of substances, and it perhaps doesn't have the health and safety risks that UV light does. So when we compared it to ATP testing, we found that in areas that had fluorescent substances, there was more ATP generally, and this is statistically different quantities of ATP compared to control areas, which we took as being adjacent non-fluorescing areas on the same item.
Mm-hmm.
Now, it's perhaps unsurprising because when you're swabbing something that you can see, you know that you're recovering a substance. Whereas, and my colleague that sort of allowed me into his hospitals would say, we say it's a bit like battleships, the ATP testing, because we're taking random swabs in the area of high touch points, for instance.
Yeah, the game battleships. I know what you mean. Yes, I mean, the swab is only as good as the bit you touch, isn't it? That's right.
Absolutely, yeah. So it was, and it's instant feedback. I mean, it doesn't work on all surfaces. If the surface is fluorescent itself, or the surface absorbs the light that you're shining upon it, unless you've got something that's highly fluorescent, it could be challenging to see. So it's certainly not going to answer all cleaning, issues with visual inspections. But statistically, it provided higher levels than, as I say, areas that didn't fluoresce, which we attribute to the fact that you're picking something up.
Okay. I mean, I take it then, Emmanuel, you're involved in looking at, was that microbiologically significant? Would that be right?
Yes.
So how does that work then? Because you can swab an area with ATP and get some sugar, and you'll get a reading that's off the scale. But actually, microbiologically, it's not really that much of an issue. What are you finding with using the fluorescence and the microbiological findings?
Thank you once again, Martin, and Sarah, for bringing me on to this podcast. What we did was to get some swabs, and then wherever the surface fluorescence, Sarah will swab that surface, and then we put it. This is a swab with a transport medium, which will support the growth of the microorganisms that have been picked from the surfaces, and that this is just to preserve the microorganism before we analyze in the laboratory, because we factor in the distance for the hospital where the sample was taken to the laboratory where we will start analyzing that. And once we get the sample, what we did was to put them out on a plate, that is the media. We have different type of media, enriched media, which will support the growth of virtually most aerobes microorganisms. So we use different type of agar. One of them was chromogenic agar, and this chromogenic agar again can be divided into MRSA. that is to isolate MRSA. We have the ESBL, we have the CPE. These are resistance, bacteria detecting agar plates. So we plate all of them, we incubate them, then the following day we look at them. The only limitation for this method for now, was we couldn't process for the anaerobic organisms. We only look for the aerobic organisms. And obviously, most of these resistance microorganisms, MRSA, ESBL, and when we look at them macroscopically, that is looking at the colonial or morphological characteristics on the plate the following day, we saw some growth. Again, that's supposed not to be from a clean surface, because before Sarah took the sample, the hospital cleaning protocol said that they check all the list that this surface is clean, everything is fine. But when she applied touch, she was able to see some fluorescence, and that is a kind of, not very clean, but it could be a dead matter or live or microorganisms. So to cut the long short, we did the morphological analysis, we did the phenotypic analysis, and then we finally sent it for discriminatory identification. And this is where we use Morditov. We don't have Morditov in our university, but being a practitioner, so I have a collaboration with my colleague in the NHS hospital. So I took the isolate to them, and they were able to identify, lo and behold, the surface that was deemed as clean according to the NHS cleaning protocol. We were able to isolate Staphylococcus aureus, so which is a very debilitating microorganism.
Yeah.
And there was another one that we isolated. It was a gram-negative, and it's called Leliota amnagneta. So this is again not supposed to be where patients are being treated in the hospital. I think one of them, if I'm not mistaken, Sarah, was from the theater, one of those samples where you got the positive from. Yeah, so that is the microbiology aspect of the analysis into that.
Yeah, because the problem with ATP is the organism can be dead and you'll still get a high ATP reading. So that's the issue.
Yeah.
Can I talk about the practicality, Sarah, of using the light? Because the one I used, you had to turn the room lights out, and and you had to wear special goggles while you're using it. So the one you're using, can you use it in normal daylight, but in that sort of thing without safety goggles, the blue light?
I'd obviously have to understand the specification of what you used before, but those that we use fall into what's called a class 2 category, which means that their use around people, there's a hazard distance to them, so you certainly wouldn't look into the light because of the intensity, but your eyes should blink should you be accidentally exposed. The purpose of the goggles, it does have a health and safety purpose, but also you wouldn't see any fluorescence without them if you were using blue light.
Okay.
So the UV is certainly a protective element, but for the blue light, you need those goggles to be able to see.
So I take it then you wouldn't be using these pieces of equipment doing an audit with patients present in the area. You do it after cleaning's taken place in an isolation room or in an area with no patients present. Would that be right?
We've used them within occupied wards, but certainly if somebody was in their bed, and not going to necessarily want somebody coming around with a light, and so generally, we're going into bays that are being audited, which are generally in those audits that I attended. They weren't auditing spaces where there were patients anyway. They'd audit a bay that was, not occupied, for example, or items would be taken into a side room, for instance. So we were able to deploy it quite safely. And the members of the public that saw us using it would be very sort of, what are you doing? And quite interested, I think. And we did have some feedback of people saying it's nice to see these sort of investments into cleaning the environment, essentially.
Yeah. I mean, the problem with microbiological sampling is actually we don't really know what the optimal method for sampling the environment is, and most hospital laboratories are not accredited to sample the environment anyway. They'll have a go, but they're not able to do it, in a reliable manner, because there is no standard method of doing it, and often you have to use different enrichment techniques for different organisms, and so and the results come back days later where nobody's particularly interested. ATP is a little bit more useful, I find, because you can give a number straight away and people go, oh, and we used to do that on our commodes, probably like the hospital you've been dealing with. But this, again, it's only as good as the bit you touch, but presumably you're able to sweep over an area and just have a look at what's around. Does it determine the difference between different types of body fluids, as a matter of interest, between urine, say, and other body fluids?
No, I mean, sometimes there's telltale markers, and it might be that you are, and I mean that in terms of some body fluids generally fluoresce more than others, but because it may be dependent on sort of what you've eaten and therefore what you're excreting and what is fluorescing,
Okay.
the location of it could be telltale. For instance, we looked at lots of commodes, so
Yeah.
yeah, it's
Yeah, well,
that might
as my experience, body fluids can appear almost anywhere because people will put them in various different places. What about the cost of this form of technology then? Because ATP isn't necessarily cheap. The swabs don't last that long. You've got to buy the equipment, you've got to maintain the equipment, and you've got to have the time to go out and do it, and possibly some software to analyze it. Presumably, this is the price of a torch, again, plus the time of somebody doing it. Did you manage to look at cost comparisons between doing the audit in different ways?
Over my career, I've used lots of forensic light sources, and those that range from in power and therefore cost. Once you've purchased a light, because they're LED technology now, so the lifespan of the LEDs, you should be able to get years of use from them, and they're designed to be used in operational settings, so that if you drop them, they should survive a few knocks and scuffs. But yeah, it's that initial investment, really. I didn't do a sort of cost against ATP. I think we're still at that sort of the infancy of the
Yeah, I mean,
method.
you're doing proof of
Um,
concept work, aren't you? Yeah,
exactly. Yeah.
yeah. But it's certainly of interest. I mean, so what's the price of a torch, roughly?
Between sort of £1,500 and £3,000.
Okay, so that's
Or is the commercial price at the moment.
yeah. But
Um,
presumably, if every hospital in the country suddenly thought, oh, we're actually quite interested in this, there's a lot more hospitals than there possibly are forensic departments, and there may be some cost movement should it become a bit more mainstream. So, but that's not completely out with the cost of ATP equipment and the ongoing cost of the swabs.
Yeah.
True. What I would say, Martin, I think, is having that, if anybody was considering sort of using this sort of technology, to really get some advice on the specification of it and those kind of health and safety risks that we've talked through.
Yeah.
Sometimes having the most powerful instrument isn't necessary for the environment that you're actually inspecting, and you might be having unnecessary health and safety risks with it, and
Okay.
still achieve what we need to with a less powerful and slightly cheaper torch.
I mean, are you thinking of producing some sort of guidance then for hospitals? If you are thinking about this, then this is the sort of spec you might think of looking at. Are the hospital you worked with interested in going down that route as a method of assessing cleanliness?
We've been approached by NHS England, who are doing their own independent evaluation of this type of technology to see if and how it might support that kind of work.
Okay.
So that's the sort of future of it. So I believe that there'll be trials within several hospitals with their own sort of microbial testing and so forth.
Okay. That's interesting, because it's certainly, I think, a step up from just doing visual only inspections, because that gives you reassurance, but not necessarily assurance, which is a different thing, really. You and I can go into a hotel room and think this looks lovely, and then you see that little curly hair in the bath that clearly isn't yours, and you start to look closer, and it's possibly not as clean as you thought it was, really.
Yeah.
Emmanuel, from a microbiologist perspective, then, do you think this adds benefit to our current monitoring methods?
Yeah, absolutely. Because when I was still in practice, we used to do what we call environmental screen, and we get loads and loads of source. But with this kind of fluorescence, it will help us, particularly where the source does not fluorescence. You don't need to swap that,
Yeah, that's a good
because
point.
that show that, yeah. So it's only where you see fluorescence that you okay. It could be dead particles, but
Yes.
it saves a lot of money.
Yes.
Honestly, it
Yeah.
saves a lot of money.
Yeah,
Yeah.
that's a really good point, actually. It could target where you might want to do environmental monitoring, because some people do environmental monitoring,
Yeah.
and there's no point swabbing a surface where there's
Yeah.
likely to be nothing there. So I think that's really an excellent point.
Yeah.
Oh, I've going through some old papers the other day, I came across a photograph of me standing up on top of a ladder 30 years ago, sampling the top of a curtain rail dust, looking for MRSA. So it would certainly
Oh
be a
my
step
goodness.
above that. I mean, health and safety might have an issue on
Yeah.
that one there as
Yeah.
well. So yeah, fascinating. What's
Yeah.
next, Sarah? Then are you doing more work in this area?
Yes, so obviously working with Emmanuel has been transformational, really, and really understanding the sort of value of what we're finding. We have another colleague, Ben Bastaki, who's an AI expert, and what Ben is working on is whether we can automate the detection of fluorescent matter. So there is a subjective element to it. So the acuity of the person's vision, is going to really determine what it is that you see. So we're taking digital images, and then he's building a machine learning algorithm that can look to detect the difference between that sort of clean and we'll call it unclean, just for the purposes of
Yeah.
fluorescence
Yeah.
detection. Now, what's great about this is that we can feed that model with any additional information that we get. So when Emmanuel does his microbiological analysis, for example, or if we had ATP testing, we can start to feed that model to say, this is what we know about what we've seen. We know the surfaces that we're on or that we've looked at, and therefore it could offer a very powerful way of investigating an environment and learning perhaps trends within them. I mean, if we were able to start having some, should we call it presumptive analysis of, when we find stains that tend to look like this on these items, we tend to find bacteria within them. I mean, that would be wonderful. So we have got that work in the pipeline. The other thing, we have a colleague called John Firmage, who's an expert in immersive technologies. And John has been building a virtual hospital room. And so you put your headset on, you go into the room and you clean it and you say, do I think this is visually clean? And then you turn on the light. And this gives you a sort of objective metrics about sort of areas that you may not have cleaned so much. Now, it's a bit gamification, but it's quite engaging. And when we've taken it out to domestic teams,
Oh yeah.
it has been that it's something a bit different. It's
Yeah.
a different way of learning. And also, the nice thing with virtual reality is you can put people into fairly dangerous settings. So you could
Yeah.
mimic somebody's working environment to say, actually, you're working around some really horrific pathogens.
Yeah.
We've got a few exciting projects, I think.
I love that sort of thing, and actually, when you show that to people, that sort of thing to people who work in hospital cleaning, who generally are not made to feel the most valued members of staff, this actually shows them that you're interested in what they do, and what they do is important. I mean, I always think it's super important. They're the most valuable parts of the team, but it's investing in their need. Cleaning's a science at the end of the day. I mean, I'd be interested in if you could use it to work out which are the most contaminated areas of a room, because nobody really knows that, because you can't swab every surface. But say, actually, if you were going to go into a room and you can only clean 5 surfaces, these are the top 5 you've got to clean. So actually having some solid data on what are the areas you need to target. If you've only got 5 minutes to clean a room and it should take you half an hour, where do you get the biggest bang for your buck? I could see that.
Absolutely. And with the domestic staff that we've worked with as well, they get quite competitive.
Oh yeah.
I found those that have their own areas to clean
Yeah.
They're so the diligence and dedication to that.
Possessive
We had
and
more.
possessive and dedicated. Yeah.
Yeah,
Yeah.
definitely. And then the one lady was telling us how she cleans the walls, and she said her colleagues were sort of, taking the mick, saying, "What are you scrubbing those for?" Then we turned the lights on the walls, and there was this kind of,
Wow.
"Oh my
Wow.
Oh,
goodness," yeah. And yes, so it's been really well received. I've never wanted to come across to anybody as like, "You've missed a bit here," because that
no.
isn't the purpose of it at all. It's about, if you can't see something, understandably,
Yeah.
you know, but if you're showing someone that you want going, "I want using this for, for the good,
Well,
educationally."
I think you just hit the nail on the head there, because if you turn the torch on, you don't need to say you've missed a bit, because
Yeah,
the person's going to look at it and say, I've missed a bit.
indeed, Martin,
Telling
yes.
someone they've done something wrong never gives a positive result. You go
No.
through the 5 stages
Yeah.
of grief
Yeah.
and all the reasons why. But showing people so that they think for themselves, actually, I could have done better there. That's super
Yeah.
powerful. So, yeah, I
Yes.
love that.
Yeah, I think
Loads
so.
of potential for this. Loads of potential.
Yeah. Yeah.
Well,
Well, if you want
Yeah.
to come down and visit
Uh,
us, Martin, and get your VR headset on, we'll get you
Um,
scrubbing the virtual
okay,
room.
I'm going to take you up on that.
Yeah.
I'm definitely going to take you up on that.
Yeah, you are more than welcome.
Yeah. But thank you very much, both of you, Emmanuel and Sarah. I've been fascinated by this piece of work, and I think it's got legs, lots of legs, and
Thank
lots
you
of
so
potential
much.
in so many ways. So I really appreciate the time you've taken to talk to me about it, and lovely to hear your passion about it for people who are not in infection prevention space. But just goes to show that there's something in infection prevention for everybody.
Thank you.
Okay, everybody. Thanks for listening to this episode, and we'll catch you again on the next episode of Infection Control Matters.